Today we'll talk about cerebrospinal fluid analysis in horses, cattle, and sheep diagnosed with rabies, a retrospective study of 62 cases. This is out of December 2022 from the Canadian Veterinary Journal, and the link is in the description. It is open source, so we will go through the entire article. Let's start.
Brief communication. Cerebrospinal fluid analysis in horses, cattle, and sheep diagnosed with rabies, a retrospective study of 62 cases. Abstract. Objectives. This study aimed to characterize the findings in cerebral spinal fluid analysis of horses, cattle, and sheep diagnosed with rabies. Animals. The study included 62 animals diagnosed with rabies at a referral hospital. Methods. This was a retrospective study using medical records from large animals with neurological signs and confirmed positive direct immunofluorescence test for rabies from 2003 to 2020. The results of CSF analysis are presented descriptively. Results. Cerebral spinal fluid samples from 62 animals were retrospectively evaluated. Of these three species, 28% showed increased protein concentration, whereas 58% presented mononuclear pleocytosis. In total, 37% of the samples had protein concentration and total nucleated cell count within the reference range. Conclusions and clinical relevance. Cerebral spinal fluid from animals diagnosed with rabies was either normal or characterized by mild mononuclear pleocytosis and hyperproteinuria. Rabies is a severe and fatal neurologic disease that affects animals and humans. The definitive diagnosis of rabies is confirmed post-mortem. The gold standard test is direct immunofluorescence antibody on central nervous tissue, a PCR, mouse inoculation test, and indirect immunofluorescence of brain tissue can also be performed. As the clinical signs of animals with rabies are non-specific ante-mortem tests that support the diagnosis would be of great value. A cerebral spinal fluid cytological analysis provides valuable information that can help condense and prioritize the list of differential diagnoses and aid in the establishment of treatment and prognosis. To our knowledge, current data on changes in the CSF caused by the rabies virus is limited, but mononuclear/lymphocytic pleocytosis seems to be the predominant finding. However, the degree of pleocytosis varies among cases. Although several techniques have been studied to develop a reliable ante-mortem test for rabies in other species, especially humans, these methodologies have not yet been extrapolated to large animal medicine. Improving the current knowledge of the CSF characteristics of large animals with rabies, therefore, could be of value to guide medical decisions, especially due to the zoonotic risk associated with rabies infection. The aim of this study was to retrospectively describe CSF cytology results from horses, cattle, and sheep with a confirmed rabies diagnosis. Our hypothesis is that the results would show a mild mononuclear/lymphocytic pleocytosis. Medical records from the large animal clinic at the São Paulo State University were reviewed. The inclusion criteria were any horse, cattle, sheep, or goat, regardless of sex or age, with a post-mortem positive diagnosis of rabies. Animals without CSF collected or with a CSF red blood count 2,000 cells/milliliters were excluded. Although it has been reported that RBCC 13,200 per milliliter does not significantly alter protein concentration and total nuclear cell count, a lower limit was used to limit any microscopic or macroscopic influence. The following information was collected from each medical record: species, breed, age, sex, days of evolution until hospitalization, and the CSF analysis. The CSF was collected aseptically from the atlanto-occipital and or lumbosacral space using sterile spinal needles. Samples were placed in sterile glass tubes for analysis within 1H. The CSF analysis was performed as previously described. Analysis consisted of an initial macroscopic evaluation for color and clarity. Protein determination was performed using a biochemical kit. The RBCC and TNCC were conducted in a Neubauer's chamber. Differential cell counts were obtained through centrifugation. The cells were stained with Diff-Quick and the cell differential was performed on a 10,003 oil objective. The normal CSF reference ranges of this report were based on previously published data. Normal TNCC range was 0 to 6, 0 to 3, and 0 to 5 for horses, cattle, and sheep, respectively. The protein concentration was considered normal between 5 to 100, 20 to 40, and 8 to 70 for horses, cattle, and sheep, respectively. The results are reported descriptively and as medians and ranges. The graph was generated using Microsoft Excel 16.54. The medical records search identified 62 animals that fulfill the inclusion criteria. No goats were diagnosed with rabies during this period. In animals with multiple CSF sample collections, the results of the first sample obtained were included in the study. 4 cattle and 1 sheep had AO and LS collection performed within the same hour. Both samples were included in the study. The total number of CSF cytological analyses included in this study was 67. Of the 31 horses, the median age was 20 mo. 12 animals were mixed breed, followed by 9 American quarter horses, 7 Manga Larga, 1 thoroughbred, 1 Argentinian trotter, and 1 American miniature horse. Information regarding the clinical signs before hospitalization was available for 27 horses. The first clinical signs were observed a median of 2D prior to presentation. The CSF collection was performed at a median of 1D after hospitalization. Of the 31 equine CSF samples assessed, 6 were collected from the AO space and 2 from the LS space. The remaining 23 samples did not have a location of sample collection specified in the record. In total, 58% were clear, and 42% were xanthochromic. The median protein concentration was 51.7 mg per deciliter. The TNCC median was 20 cells per milliliter. The RBCC median was 81 cells per milliliter. In all samples, mononuclear cells were predominant. Only 7% of the samples had a protein concentration above the reference ranges, whereas 39% of the samples had a pleocytosis. The protein concentration result was not available for 2 horses. 24 cattle were included in this study. The age was available for 23 animals and the median was 1Y. 14 animals were mixed breed, followed by 5 Nellore, 2 Angus, 1 Wagyu, 1 Holstein, and 1 Jersey. Historical information was available for 23 cattle. Clinical signs were present for a median of 2D before presentation. The median number of days after presentation that the CSF sample was collected was 1D. In total, 28 CSF samples from 24 cattle were included. 4 cattle had AO and LS CSF samples included. Of all samples, 4 were collected from the AO space and 7 from the LS space. The remaining 17 samples did not have a location of sample collection specified in the record. Only 1 out of 28 samples was sent to chromic, whereas all other ones were clear. The median protein concentration was 43.3 mg per deciliter. The median TNCC was 9 cells per milliliter. The median RBCC was 3 cells per milliliter. In all samples, the predominant cell type was mononuclear and 71% of the samples presented pleocytosis. 50% of the samples had increased protein concentrations, and 71% of the samples had pleocytosis. The protein concentration result was not available for 2 cattle. 7 sheep, with a median age of 23 mo, were included. 4 animals were Senne, followed by 2 Dorper and 1 mixed breed. Clinical signs were present for a median of 3D before presentation. After presentation, the median number of days until the CSF sample was collected was 0D. 1 sheep had both AO and LS collections performed in the same hour, resulting in a total of 8 samples included for analysis. In this study, 1 sample was collected from the AO space, 4 samples at the LS space, and 3 samples had no site of collection noted in the records. None of the samples were sent to chromic. The protein concentration was increased in 57%. All samples presented pleocytosis. The RBCC median was 13 cells per milliliter. In all samples, mononuclear cells predominated. 57% of the samples presented with an increased protein concentration, and all samples had a pleocytosis. Considering all samples, from the 3 species in this report, 30% had an increased protein concentration, whereas 58% had a mononuclear pleocytosis. 26 samples had protein concentrations and total nucleated cell counts within the reference range. 5 animals had CSF collected from both the AO and the LS spaces on the same day for comparison. Out of the 5 AO samples, 3 had increased TNCC, and 1 had increased protein concentration. Considering the LS samples, 3 had increased TNCC and 3 had increased protein. Interestingly, out of the 39 samples that had a pleocytosis, only 17 presented concomitant increased protein levels. Out of the 19 samples with increased protein, 17 also had concomitant pleocytosis. This report describes that CSF cytological analysis of horses, cattle, and sheep with rabies may either present with mild mononuclear pleocytosis or be within the reference range. This finding is congruent to previously published case reports and case series. In this group of cases, the CSF neutrophil count was mildly to moderately increased in a few cases. However, it was never the predominant cell type. This helps differentiate rabies cases from neurological diseases that cause marked suppurative inflammation, such as bacterial meningitis, and diseases that cause marked non-suppurative inflammation, such as bovine herpes virus 5 meningoencephalitis in cattle. Solely based on CSF results, it might be difficult to differentiate rabies from other viral neurological diseases in equids, such as arbovirus diseases, as most also present with a mild to moderate mononuclear pleocytosis, and from leukoencephalomalacia, which presents with a mixed or mononuclear pleocytosis. One of the main limitations of this study is the lack of information regarding the CSF site collection for 43 samples. In the 5 cases from which CSF was collected from both the AO and LS sites in the study, the TNCC was similar. In contrast, the protein concentration differed and was higher in 3 LS samples and 1 AO sample, respectively. Although the protein concentration was increased in LS compared to the AO samples in animals from which CSF was collected from both sites, mononuclear pleocytosis was the predominant finding in both samples, suggesting that cellular composition is less affected by sampling site than total protein concentration. Due to the small sample size, it was not possible to differentiate species-specific changes. However, the 3 species appear to demonstrate a similar pattern of abnormality. The only abnormality that was different among species was the high percentage of xanthochromic CSF in horses compared to cattle and sheep. The median RBCC was also higher in horses than in cattle and sheep. If this change truly represents a specific equine response to the rabies virus, or is just a coincidence, remains undetermined. As the classification of color is a subjective evaluation and may present inter-individual variability, this information should be considered carefully before definitive conclusions are made. In addition, no predilection for breed and age was detected in any of the 3 species. The number of females and males were evenly distributed in horses and cattle but not in sheep. This difference is believed to be due to the fact female sheep were overrepresented in the hospital's caseload compared to male sheep. Another limitation of this study is its retrospective nature, allowing for misclassification bias and confounding. As the sample was recruited by convenience, it might not represent the general population of animals with rabies in the field. Lastly, our small sample size limited the power of the study and the possibility of statistical analysis. In conclusion, the study describes that CSF cytology of horses, cattle, and sheep with rabies is likely to have mild mononuclear pleocytosis or might be within the reference range. This result is especially important as there are currently no reliable antemortem tests for rabies available for large animals. Further studies with additional parameters and molecular identifications are warranted.
So overall, this is a fairly straightforward article. I think one of the most important aspects to take from this that wasn't included in the abstract is the neutrophil count, where they had stated it was elevated in some samples but was never the predominant cell type. So if you have a patient to where you collect a CSF and the predominant cell type is neutrophils, then it seems less likely to be rabies. And the example they gave here, where it's more likely to be rabies, is the bovine herpes virus. But other than that, I don't have much to add. I think the abstract did a really good job summarizing everything. Looking at the figure, not much differences between the three species provided here. I thought it was interesting no goats were included in this study. I know goats can definitely get rabies, so this is likely just by luck that there were no goats included. Since none were included, we can't say for certain if this holds true for goats. Generally, sheep and goats share a lot of similarities, so I would assume this would hold true, especially with the neutrophil count, and that a normal CSF does not rule out rabies. Overall, it was an interesting paper with good, straightforward information. That's all I have. Thank you.